Yes, as Becky said, I'm a professor of biology at Florida Southwestern State College. I work specifically at the Collier campus, so I'm here in Naples in the area. I'm a Southwest Florida boy. I was born in Fort Myers, and I've been here the most of my life. So I'm very happy to be able to work down here full time and not only teach school and teach biology, but also work in the field that I've grown up loving, and that is, of course, the field of herpetology, which is the study of reptiles and amphibians. And I've always had a really, really soft spot in my heart for turtles of all species, but box turtles have consumed the majority of my life for the last 2.5, almost 3 years. We've done a lot of research, and I see that several of my students are actually attending this lecture, so that's really cool. So they'll get a shout out here at the end, which is awesome. And without these students, none of this work would be possible. So I always like to shout them out for all of their hard work. But we're just going to kind of go through, talk a little bit about box turtles, and then update you on the status of the project, specifically focusing on the Naples Preserve and what we've learned. But anyways, the first thing I always like to go over is, what actually is a box turtle? A lot of times that term box turtle is just kind of thrown around for any turtle that people see on land crossing the road, but it's actually a very specific group of turtles that are considered box turtles. So first and foremost, there's between 9 to 10 extinct species or subspecies in the genus Terrapinae. That's the scientific genus that box turtles are in. And the United States is where a large majority of those species and subspecies are found. This is a lovely range map of said species. And some of you who may be from up north, you probably grew up seeing the true eastern box turtle, Terrapini carolina, which kind of makes up this orange swath of the map. Goes all the way up to the northeastern United States. They kind of go west a little bit into Tennessee, Kentucky area. But once you kind of get over in this region, in the true west, kind of near the Mississippi areas, you start seeing this other green swath, and that's indicative of another species or subspecies, depending on how you talk to, popping up, called the three-toed box turtle. And as that goes west, and you get some other species like the ornate box turtle, the Mexican box turtle, the desert box turtle. All those species kind of pop out that way. And you also notice there's kind of this little purple swath over here at the Panhandle, Florida. We get another species in Florida called the Gulf Coast box turtle, which is mostly in North Florida. But as you can see, most of the Florida peninsula is actually covered by yellow, which is indicative of the species we're going to talk about today, the Florida box turtle. And this is just a picture from a group known as Garden State Tortoises, just showing the diversity of the color patterns that all of these species and subspecies of box turtles have. We've got a Gulf Coast box turtle, we've got a Florida box turtle, we've got a three-toed box turtle, we've got this is what's called a Coahuilan box turtle, which is a Mexican species. We've got ornate and desert box turtles there. So much variation across the genus and even within the species, which is pretty cool. They get the name box turtle for a reason. A lot of people will just call, like I said, just call any turtle they see on land a box turtle, but that's not the case. Box turtles are very specific because they have what's called shell kinesis. Kinesis means the ability to move their shell, and they have these hinges down on the bottom part of their shell, which can close up like the flaps of a box. That's where the name box turtle comes from. So they can actually completely box themselves up. And this allows them to have great results in terms of surviving a lot of predators. There's not a whole lot of predators that can really get into and completely damage and feed on box turtles. There are some, of course, some of the bigger mammalian predators can do that. And when they're small, they don't really have the full hinge or the hard shell to protect themselves. But as evidence in the preserve, there's a lot of these box turtles that have teeth marks and chunks taken out of their shell that are still living probably decades after their first encounter with a predator in some cases, because they have such a great protective strategy with this boxing behavior, which is really neat. They exploit a lot of different habitats. These turtles are amazing. When you look across the range, they live in desert prairie, they live in true desert, they live in swamps, they live in coastal mangrove estuaries, they live in shell hash islands, they live in pine flatwoods. They can live pretty much anywhere. The biggest issues that face them is not the habitat that's available, it's when humans come in and sort of develop things, as we'll talk about in a second. These animals, unlike the gopher tortoise, well, I guess Becky might tell you otherwise, that gopher gopher tortoises are a little bit of opportunistic omnivores as well. They're willing to feed on meat items, but we typically think of gopher tortoises mostly herbivorous, feeding on plant matter and things like that, even though they will feed on other things. But these turtles are what we call true opportunistic omnivores. If there's something that's available that smells good to them, they're probably going to eat it. And sometimes that is as cool as like a little slug that we found one of our radio tag turtles at the preserve feeding on a couple months ago. It could be something as interesting as the feces of other animals. We actually found one of our turtles feeding on coyote feces at another site, and one feeding on raccoon feces as well. So they will eat on whatever they can find. Varies. We find seeds in their diets. I've got a couple of students working on a dietary study looking at what kind of food is found in their fecal samples. They eat pretty much everything: bugs, poop, meat, whatever it is, you name it. If they come across it, they're going to eat it. Now, in terms of our particular study, someone have a question? Species we're going to focus on, of course, is the Florida box turtle, which, as you can see here in this figure, occurs throughout most of the peninsula of Florida. Once you get kind of up towards the Panhandle area, you get this what we call an area of uncertainty, where the Gulf Coast box turtle comes in. There might be some integrating between those and the Florida box turtle. You also get some influence from true eastern box turtles up in northern Florida. So there's kind of some mixture between these. And so a lot of people consider these three to be subspecies of one another. They're all the same species, but different subpopulations. But there's actually evidence that supports that the Florida box turtle may actually not be a subspecies of the eastern box turtle. It may actually be its own species, which has huge impacts on conservation decisions, as FWC is approaching making some decisions on the conservation status of this species, even as we talk here, which is really a good thing. Considering some of the threats. And this is what that classic Florida box turtle looks like. As you can see, they've got this sort of yellow striped shell. There's a lot of variability in what the bottom of their shell looks like and their facial patterns, but in general, this is what the shell is going to look like for the most part in Florida box turtles. And they're an extremely beautiful species, and that's kind of what gets them into trouble in some cases. As I mentioned, with box turtles overall, they have very diverse habitat use, but Florida box turtles, to an extreme degree, have variable habitat use. Like I said, they'll use flatwoods with a lot of pines, saw palmetto all over the place. They'll use coastal scrub areas, basically on beach dunes. They'll use mangrove forests, which are filled with water. So these animals, even though they're considered a land turtle, are very, very aquatic and can be found swimming in a lot of places. Even at the preserve, when it rains really heavily, we find a lot of them swimming in flooded gopher tortoise burrows or in really deep spots. They are not afraid to get in the water, even though they're considered a land turtle. Here's a couple we found breeding in the mangroves, which is pretty cool. Now, in terms of threats for this species, they are of conservation concern because of a lot of reasons, and the most damning reason always is habitat destruction. Florida has a greater population growth than many of the other states in the southeast, and that leads to more development, more destruction of natural habitat, more building of roads, and these animals don't do great when roads go through their habitat. When roads are built through, that brings traffic, and that brings people that aren't paying attention. And when they're not paying attention, you may smack a turtle that's crossing the road. As you can see, here's one that was actually getting ready to cross the track of an ATV four-wheeler track. So going right through heavily trafficked areas is not good for the health of this species. This cuts population sizes down. It cuts genetic diversity of species, and it causes a whole host of problems. So habitat development and destruction, of course, are kind of one of the leading causes for decline in the species across its range. Now, poaching is also a huge concern. As you can tell, these are a very beautiful turtle, very, very pretty species, very beautiful yellow markings all over the place. They don't get very big. You know, the biggest females usually get around this size, less than the size of a football. So they are very wanted in the pet trade. You can legally own up to 2 turtles or 2 box turtles per person here in Florida. So if you had a household of 4, you could have 8 box turtles as pets. If you were just a single individual, you could have 2 box turtles as pets. And I don't necessarily see the issue with just having, a couple of pet box turtles. The issue is that The demand for pet box turtles leads to exploitation of box turtle populations. This was a poaching event that occurred October 2019, where they found thousands of turtles, several hundred of them box turtles, in the house of a man over in Lee County, who was catching them from throughout the Southwest Florida and then shipping them over to China for the pet trade. And it's not just China that demands a pet trade; these are still in demand in the United States. It's just that China uh, pays a very, very pretty penny for a lot of these species. Thankfully, they were able to get a lot of these animals out, and they were able to release them. I was able to participate in some of the release efforts of these animals, marking, tagging, and getting measurements and taking genetic samples on them, so we can, in the future, have a better grasp at the best ways to support these big confiscations and things like that. So that is always something we're cognizant about, is not revealing too much about the areas that these animals we find when we do research to prevent poaching and things of that nature. Disease is also kind of on the forefront. There are several diseases that can spread between turtles. Some of them you may be familiar with gopher tortoises, known as mycoplasma, that causes their upper respiratory disease, where they kind of get a snotty, bubbly nose. That can occur in box turtles as well. There's also a lovely virus going around called the ranavirus. And if anything, we're all pretty much virus experts after this last year. We know how fast viruses can spread, especially respiratory viruses. Ranavirus is really bad, though, because it not only causes potential respiratory distress, it also causes a necrosis, causes the flesh around their face to start basically peeling off, and there's no cure for it. So if an animal contracts ranavirus, it's going to die most likely. Even if it has it sitting latent in the system, if it expresses, most of those animals die from ranavirus infection, which is very sad. And a lot of that is involved, again, in that habitat destruction. When home ranges get cut, that squishes more turtles together. When they're squished closer together, they can spread disease a lot faster, which is a very sad thing. So that's why they're an important species to study, to understand home range and to understand what their habitat use is. And that kind of leads us directly into the research that we're doing here. I'm going to show you a nice collage of all of these different individuals that we found at a couple of different sites. But our study, we work with many different agencies. It's not only just our college doing the research. We work directly with the Florida Fish and Wildlife Conservation Commission. They funded some of our research. We work directly with their biologists, documenting what we see. If we see anything weird or not normal, we report it to them. And that's why monitoring populations this way is so important. When the biologist can dictate there's something astray with a population, we can report that to the commission. That can allow for conservation to occur, which is great. We're also working directly with organizations like the Tangled Bank Conservatory, and this is like a lovely organization that is doing genetic mapping of box turtles across their range to specifically figure out best places to release animals that are confiscated in big poaching busts, like I showed you before. So if we know what genetics match which parts of the state, that can better help us to release some of these animals in places they belong, so we don't kind of mess up the genetic integrity of populations. So that's where the comparing of genetics. We're also doing some cool work where we're comparing the phenotypes, or the physical variation in the color patterns across populations. We're finding that in some populations there is very common patterns that aren't found in others. So we're going to do some work with a colleague of mine up at Indiana State University. She's kind of the guru when it comes to differing color patterns in turtles. So we're going to do some really cool analysis to look and see if there's maybe like an adaptive trait towards the evolution of these animals based on the color that they develop. One of my big things is I'm a trained reproductive biologist, specifically an endocrinologist when it comes to turtles. So I'm all about studying reproductive effort, when they reproduce, how much they reproduce. So that's one of the big kind of sub projects of this overall study, as I'm looking at the reproductive timing and output of these animals. And we've learned some really, really cool stuff that I'll share with you later. And one of the big things that we're focusing on at the Naples Preserve, and what I'm going to largely focus on in this talk, is the looking at the home ranges of these animals, how wide their ranges are, what habitats they use. Is there a difference between different seasons? Is there a difference between males and females? So on and so forth. And that's kind of going to be the big take home that we go through today. Now, the specific questions that we're asking, of course, are: is there variation in seasonal activity? here in Southwest Florida, we don't really have winter, right? We've got summer and slightly less hot summer, pretty much. Those are our main seasons. But we kind of break our seasons down into two: a wet season and a dry season based on average rainfall. Wet season usually starts, our dry season usually starts around, I think, like mid-October, and then wet season kind of picks up in April, May. So we split our two different seasons in terms of tracking data to see if there was a difference in the home ranges based on the seasonal patterns. Now, we also looked at what habitat types they were using. As you can see, the preserve has several different microhabitats within it, and we looked to see if there was one that was maybe used more based on the number of times we located an individual in it. We also compared males versus females to see if there was a difference in home range. Historically, males usually have a much larger home range than females in box turtles because they're always out looking for some ladies, so they will travel several miles sometimes to find a lady so that they can reproduce. Preserve itself obviously isn't miles wide, so that may influence how much the home range varies between these. We also wanted to look at the influence of gopher tortoises on these animals. We know gopher tortoises, the classic statistic is that there's over 300 different species that have been documented using gopher tortoise burrows, whether they're commensals or mutualistic species. We wanted to see if box turtles were really kind of one of those things. So we were hoping to look at burrow interactions of box turtles with gopher tortoises and the like. And then, of course, as I mentioned, understanding the reproductive ecology of these animals in this southern part of the range is really important. And in this site specifically, we found some really cool stuff that I'll report to you today. The methods overall that we used, obviously, a lot of our data is coming from the Naples Preserve, which if you're in this talk, you're probably all pretty familiar with. It's a recipient site for gopher tortoises, even though it's no longer really a recipient site for gopher tortoises because the population's maxed out. As Becky has told me, there's approximately or over 150 tortoises over the 9 acres that are here. So there's a lot of tortoises, very dense. There's lots of burrows here. So that means there's lots of microhabitat that's available for box turtles to potentially use because of all the burrows. That the habitat of the Naples Preserve, as you can see here, is separated into 4 kind of sub-habitat types. We've got the oak rosemary scrub, which is kind of this thick area, again, dominated by a lot of oak species as well as rosemary plants. There's also mahogany in some of these areas. We've also got pine flatwoods, which is going to be dominated by the pine trees that you may see, as well as the solid palmetto. And then you've got this sort of grassy open meadow area that does have some little oak rosemary patches growing within it, but it's kind of designated by this blue stem grass field, things like that. And then over here, before the preserve, was officially kind of fenced in and built in these neighborhoods, there used to be kind of a wetland area as well. And you can kind of see the residual impact of that because of some of the vegetation that's able to grow over there and how well it kind of floods in this area during the wet season. So we documented that as the former wetland as well. Now, as you may know, the Naples Preserve is kind of found smack dab in the middle of a neighborhood and busy street. These habitats typically in nature benefit from fire usage. So natural fire started by lightning or also controlled burns to burn things and thin out vegetation. And because the proximity to these areas can't be managed by fire. So poor Becky and her student army, they go out and they mechanically manage it. They go out and they cut down trees, they rip out vines, and maybe some of you have been participants in that to help manage the habitat and make it as close to natural as it can possibly be without fire. And if you haven't seen it in the last couple of months, it just absolutely looks fantastic. Some of the areas they've cleared out, it looks like a burn went through without the blackening of the tree bark. It's fantastic how well they've cleared it out. And we know the turtles and the tortoises likely very much so appreciate that when they get some of this undergrowth cleared out and allows for new growth to occur. When it comes to collecting data, again, we're focusing largely on radiotelemetry here. So we fixed about 10 box turtles with radio transmitters. We used a couple of slightly different types of transmitters, but overall they had about 400 days of radiotelemetry use, so they could be tracked for 400 days. Plus, some of them got this kind of gooey epoxy, which solidified as a clear epoxy, and then later on we switched to a marine epoxy just because of how how wet these animals were getting. We found because this lasts a little bit longer in humidity and wetness, and we had to paint this to kind of to make it a little more blending color because it's a nice big white epoxy compared to the clear see-through stuff. But it allowed us to monitor these animals for over a year. We actually got about 13 months of data from our longest turtle in this study, and we were able to get some really cool information, as you'll see. We tracked turtles times per week, and when I say we, I said sometimes I track turtles once a week, or I would tell my students to track a couple times a week. Like I said, without the students, this project wouldn't have been anywhere near as successful as it was. So about 2 times a week. The times were kind of randomly staggered. Sometimes it'd be the morning, sometimes it'd be the afternoon, obviously pending weather and things of that nature. When we did find a turtle, or at least got a really strong GPS signal from the turtle, sometimes we couldn't find them because they were busy, sometimes buried over a foot underground, which is really cool. We would take a GPS point with a little app on our phone, which was really cool. Not having to buy a big, expensive satellite GPS tracker, we would just take a GPS on an app known as the GPS Tracks app. On our phones. We would document the habitat type. Was it flatwoods? Was it scrub? Was it prairie? Whatever it may be. We document the behavior. Were they breeding? Were they eating? Were they hiding? Were they out in the open but not moving? As well as the temperature and humidity right around the area that we found or hypothesized that the turtle would be found in. Just kind of gives us a better idea of the microhabitats, temperature and humidity compared to the rest. And this is what it looks like when a student would go out and actually find a box turtle. We didn't handle them every time that we found box turtles, but we wanted to check the integrity of the tags every so often too, because sometimes tags fall off of turtles. So we want to make sure the epoxy held up. And luckily that never happened at the preserve site. One of our other sites, we actually did have a turtle's transmitter fall off, but luckily one of our students actually tripped over that turtle and found the turtle at the same time, right after the tag had fallen off. So we were able to reattach that tag. And I think Christina is in here. So sorry to call you out, Christina, but it's a good reason. It's a good thing that you tripped over that turtle and found it. So we were able to get the tag back on top of it, which was great. And when I say we would record GPS points and things like that, often we would be me standing way back and sending students into the thick bushes to actually take the GPS points while I sit back there and take on my professor status and say, oh yeah, you're doing a great job. So here's a couple of students stuck in a really dense patch of vegetation getting information on said turtle. Perks of being a professor, right? Now, when we got all of this data, we did a couple of different analyses. We actually have a collaborator at NOAA by the name of Dr. Daniel Hoking, and Daniel is a wizard when it comes to analyzing data. He uses a open source program called R, which I have no idea how to use, but he's a wizard with it, so he can do a bunch of complex statistics. And I won't bore you with the overall idea of those, but he's really good at it. And he also uses a program called QGIS, which is a geographic imaging software where they're able to actually look at our GPS points that we took and then provide analyses based on the space that we found them. And some of those analyses include things called minimum convex polygons. And you'll see what these are in just a moment. But a minimum convex polygon, its main job is to effectively show us the range that an animal has traveled. So it takes the most peripheral points on the outside of a general area that the animal was found, and it connects them at angles 180 degrees or less, and it gives you an estimate of the smallest amount of home range that an animal has covered, and you'll see what that looks like momentarily. We also use another method that are called kernel density estimates, and what these estimates do, they're really cool. They're kind of new to me. But they basically take the 90% probability that an animal will be in an area based on the information we've documented. So it takes all of the points from that animal that we've documented over the year, and it estimates the likelihood of that animal spending time in a specific region up to 90% accuracy, which is really kind of cool. So you kind of see what that looks like. And this is a cool model that we can kind of use when we're doing future surveys for these animals at the preserve or other places. Based on these probabilities, we can say, okay, we know they're more likely to be in these areas as opposed to these areas, so we can focus our efforts in a very specific region, which is neat. And then we will calculate the home ranges from these methods in hectares and square hectares and how far they may have traveled and what habitat they may use. And we also calculate just how many times we found them in flatwoods, how many times we found them in rosemary scrub, etc. And I see some people kind of looking like, what's going on in this picture? Well, this picture is kind of interesting. You can definitely see the one turtle up here, right? This is number 20. His name is Lou. And Lou actually was taking some really good advantage of a flooding event because there's actually a female turtle under the water here as well. So he was using his buoyancy to help position himself to reproduce. And that was a common occurrence that we saw in this population, especially with Lou. Lou was found macking with several ladies out in the preserve, at least 3 different ladies we found him reproducing with. So he was doing his best job at spreading his genetics amongst the population. So I think we found, this is actually number 70. These are 2 radio tag turtles. This is Blue and Missy hooked together here. We found Lou with Priscilla, and we found Lou with number 5, and I can't remember what her name is, but she wasn't a radio tagged individual, but we found him with at least 3 different ladies at some point in time throughout our study. So he did a really good job at passing his genetics on. Good job, Lou. Now, I'm going to kind of give you the preliminary results of things, try to break them down into nice bite-sized bits so you can kind of see the big picture. But in terms of habitat preference, we had over 1,018 encounters with 10 individuals over the year, 13 months of study that we did. That's a lot of time seeing turtles, which is really, really cool. With the 10 individuals that we found, so we had 5 males and 5 females that were both radio tagged. So this includes both of those. But 38.6% of those 1,018 encounters were in the oak rosemary scrub border habitat. So it's a pretty good amount of time that we saw these animals using oak rosemary scrub. All right, and this is what kind of like an oak rosemary scrub island would look like in the preserve if you haven't been back there. And believe it or not, there's actually a turtle in this picture. Anybody pick out the turtle? Adrian, you can't, you cheat. I think you were there for this one, so. Right, if you can't pick it out, here's a zoom in, right? And the red highlights this turtle right there. That's Lou, number 20. And I highlighted another sucker. There's actually a black racer just kind of hanging out in the same habitat with them. But you can see there's a lot of oak canopy cover in these spots, providing them a lot of sort of escape from the sun. And there's lots of good like detritus and mushroom growth underneath these areas. And we found them eating some mushrooms at times underneath these spots. The other habitat that they were found in actually the most was the pine flatwoods. They really liked the pine flatwoods habitat or at the border of those habitats at least. 47.7% of their overall captures and locations were found in pine flatwoods. And again, if you know anything about pine flatwoods, there's lots of saw palmetto. And there's another turtle in this photo. Does anybody see the turtle? And if you don't see it, I'll zoom in. It's right there. That just goes to show you how good these animals are at blending in with their habitat. And if we didn't have radio tags on them, finding them 1,018 times would be near impossible. You kind of really have to get lucky to find them, go out at specific times of the day when they're most active and hope that they're walking across your path at that point in time. Otherwise, it's really difficult to find them in some of these habitats. We found about 13.7% of their encounters were in the prairie meadow or the former wetlands combined. So they didn't spend a whole lot of time out there, but there is one individual that spent basically all of her time out there, and that's Priscilla. And there she is doing a really good job of protecting herself. So not only is she boxed up in her shell, but she's in the middle of a cactus. So if anything wanted to go in there and eat her, they would have a really hard time getting a hold of her. So she did a really good job. She's one of our favorite turtles. She was actually the first one we ever tagged in this study. Becky had been seeing these animals for many years before we started, but she's the first one we actually microchipped and measured and got data on. She's also the first one we applied a radio tag to, and one of the first ones we ever saw nesting in this population, which was really cool. So she's just she's kind of holds a special place in mine and all the students' hearts because she's just one of the coolest turtles, and she's one of the easiest to track too. So she was always a fun one to work with. And we've still got a tag on her. She's got some really cool information for us. Now, overall, the results that we're seeing, all right, so I'm going to give you this data in what we call hectares, all right, that's the measurement. So the minimum convex polygons of these animals, we're going to break them down by sex and also by season. So female turtles in the wet season had a minimum convex polygon home range of about 1.25 hectares. All right. In the dry season, that reduced a smidgen to about 1.20 hectares. And based on data analysis, there's not really a significant difference between their home range between these seasons. All right. Males, on the other hand, had a 1.66 hectare home range in the wet season compared to a 1.25 hectare home range in the dry season. So we still have to do the statistical analyses to see if that's really different. But just looking at that, it seems like males were likely on the move a lot more in the wet season compared to the dry season, which is really kind of cool information. Now, these are what all of those minimum convex polygons are all about, as I was mentioning before to you. All right. Top one up here is female. This is male, and this top line indicates the dry season. So it's kind of hard to see with the different colors, but down here, but basically each colored polygon is a different individual's home range. So for instance, this big blue one here, that's going to be number 6's home range. That's Valentine. She's was a potential drop-off turtle that just kind of showed up that Becky may have missed or may have been dropped off by somebody else at some point in time. But she had kind of a really weird home range. She spent most of her time up in this general area, but also had a big adventure all the way down to the Prairie Meadow at one point, and then over here to this east side at one point before kind of going back to her little home range over here. She's a really kind of a cool animal, but we can almost reliably every time still go back to this area and find her. That's where she spends most of her time. But this is the minimum amount of space in the preserve that she would be able to use based on those calculations. As you can see here, I mean, there's not a huge difference, but in general, the males seem to have these kind of bigger convex polygons, especially in the wet season here, taking up really big portions of the preserve, going back and forth. Our biggest adventurer here looks to be, I think it was number 20, Lou. He basically spent a lot of time in the oak rosemary scrub, but also ventured all the way up into this section over here a couple of times. And compared to the dry season, you can see those ranges are very constricted, very different compared to the wet season. So the males definitely seem to be moving a lot more in the wet seasons. And these are just those hectare estimates to show you the comparisons between each group. Females, on the other hand, you can see in general, in the dry season, they actually appear to be, at least for one individual, wider. For instance, Valentine over here actually constricted her home range during the wet season. She became a lot smaller. So whether that's indicative of when nesting starts, maybe they are more likely to stay in a specific range, or maybe they move more in the dry season because that's when nesting is occurring at its highest densities. Those are all still questions we're hoping to answer, particularly with the second part of the study. But really kind of cool images that show just how much of the preserve these animals use. If I were to overlay all of these together, it would be kind of crazy just showing how much of the preserve is in use by these animals. Now, this next one, these are the kind of the cool new toy that we're using. These are the kernel density maps. All right. So kernel densities, again, are like basically a 90% probability of how likely an animal is going to use a habitat based on a prediction from the GPS data that we've taken. So the amount of hectare space that these animals are going to be using, if for females in the wet season, according to kernel density, is about 0.75 hectares. So you notice these are smaller ranges because these don't take into account all of the peripheral points and the angles between them. These are just the likelihood and the calculation of how much range is in that likelihood, as we'll see in the images. So in the dry season, females had a little bit less home range according to kernel densities, about 0.66 hectares. Males in the wet season, about 1.2 hectares, compared to 0.52 hectares used in the dry season. So according to the kernel densities and comparing that to the minimum convex polygons, there is a big difference, very likely, between male use between wet and dry seasons. It looks like the wet season is just a much more active period for these males. And that can be seen oftentimes when it rains out of the preserve. That's when a lot of times we see these animals really out and moving a lot of the times. So this is a kernel density map for one individual female. This is number 70. Her name is Missy. We called her Missy, which is short for mistletoe, because we found her during Christmas time a couple years ago, and also because she's missing toes on her front left hand, which is kind of funny. So she's mistletoe because she's missing toes. But these dots here, these little grid lines, basically indicate the probability and the likelihood that we're going to find an individual in an area. So the more orange and yellow you get, the higher the likelihood they're going to be in that spot according to the data. So as you can see here, in the dry season, she actually spent a lot of time up in the pine flatwoods, but she also spent a good amount of time down in the prairie meadow. Over here in the wet season, kind of split between the two again, probably a higher percentage in the prairie meadow area, but also some high densities in the pine flatwoods. You can see that some of these grids kind of go overlap into like traffic and 41 as well. That doesn't mean that she actually moved out of the preserve into 41. That's just kind of one of the limits of kernel densities, is that they assume that there's no barriers in a space, and that all of the space that's in an image could potentially usable space to an animal. So that leads to some kind of overestimates of where they may spend their time at some points, and that's something we'll have to kind of discuss when we actually publish a paper on this. But overall, it still gives a really kind of good picture of where they're spending their time. Think of these as kind of like hot spots. The lighter the square, the more likely the animal is going to be there. This next one we're showing here, this is number 30. Number 30 was weird. He's a male that kind of moved, spent a lot of his time up here in the sorry, actually this is number 6. This is Valentine. She spent a lot of her time again up here in this area before moving to the prairie meadow for a lot of times. But then in the wet season, she spent predominantly all of her time up here in the flatwoods. You can just see how those yellow boxes kind of indicate that. Here's number 30. So number 30 was kind of a really weird one. He never ventured into the prairie at all. He stayed in the flatwoods completely and totally, never made a trip down to the open prairie, which is unfortunate for him because this is where a lot of the ladies like to hang out all the time. So he kind of missed out on that. He obviously had to interact because look at this density here. This is right where Valentine spends a lot of her time as well. So he had to cross over and meet her at some point. And we know that because we actually have evidence of Valentine number 6 being gravid with eggs. So she's obviously met a male somewhere up here at some point. Hopefully it was number 30. But you can see he was very patchy in the dry season, but still spent most of his time up here in these areas, but spent a lot more of it over here in the wet season. Pretty cool stuff. This one over here, this is just our good old girl Priscilla. She's another one that basically never went really into the oak rosemary scrub except for the little scrub islands in the prairie. She spent pretty much all of her life in the prairie, and I think Becky can probably confirm this as well, that I don't think she's ever seen her anywhere north of this boardwalk area. She basically spends her entire time down here in the meadow, and she's one that we find all the time because she just always hanging out in the open prairie, which is a little bit scary because we know there's a coyote visiting the preserve right now, and we don't want her to be the coyote's next meal, just because of all of the excellent data that she's provided for us, and also because, you know, we're not supposed to get attached to scientists, but obviously we're all attached because she's just the coolest little turtle. And she's given us some of the best data out here, as you'll see in a moment. But those are kernel densities and things like that. Does anybody have any questions about the convex polygons and the home ranges and things like that that I might be able to answer, or maybe able to struggle through to answer for you? I know it's a little bit of sciencey jargon, but it's really cool stuff just to show how much habitat they use. Okay, we'll move it on. All right, so back to some of those other questions. How are they using and interacting with gopher tortoises? Well, they're certainly using gopher tortoise burrows. They're not using burrows as much as we thought they would, just given the amount of burrows here. But there's at least 3 individuals that we found using burrows. This is individual number 10. She has recently been given a radio tag, so we're tracking her right now. And she's actually back in a burrow right now as of our last tracking situation. So she's living right in a burrow. The burrow she's living in right now is it seems to be an inactive one, but she is living basically right at the mouth of a burrow, which is really interesting. We had another one. It was not number 13. It was number 14, I believe, and maybe one of the students can correct me if I'm wrong. It was number 14 and number 8, a male who basically lived in an inactive burrow where there was no gopher tortoise, but had been dug by a gopher tortoise in previous years, but it lived there for like almost a week straight, sitting in this burrow, left for a few months, but then came back to that inactive burrow and spent at least another week in that burrow, making use of some hard work from a gopher tortoise. This is really great. And we've also found a couple of others using some burrows kind of toward the fringe of the edge, near the prairie and the scrub islands on the edge there, spending at least a couple days, and sometimes I visit burrows. Although I believe Adrian and I think one of my other students, Bella, they actually tracked a male using those burrows, and as they were kind of tracking him, he kind of ran out of the burrow almost to shoo them off to let them know that it was his habitat. It was kind of a really cool experience for them to see. The other big thing in terms of interactions with gopher tortoises I like to highlight is that mycoplasma. It's that upper respiratory disease that affects gopher tortoises, and we know it can also affect box turtles. And we have evidence that some of the turtles here do experience upper respiratory tract infections. It's very likely that it's mycoplasma, whether it's from the gopher tortoises or a strain that's specific to the box turtles. We don't know because we haven't tested it. But that's something I really want to look at. It's commonly shown by gooey or sort of swollen closed eyes, discharge from the nose, and things like that. In general, lethargy. So they're just usually very inactive. We found at least 3 of our turtles out here with signs of respiratory infection. One of them we did take to the wildlife clinic on Sanibel, as my former collaborators over at Crow, to look at and potentially swab for infection. But once we got him over there, all of his symptoms completely cleared up. Of course, you're only sick until you go to the doctor, and then everything clears up, so they don't believe you. So he ended up coming right back, and he didn't really have any issues. Priscilla actually had a bout with upper respiratory for about a week and a half, but we just kind of let her stay underground and fight it off herself, and then she obviously did just fine later on and cured it. We haven't seen any direct death because of upper respiratory infection, although Becky did find one of our tag turtles, not a radio tag turtles, did find one of them deceased in the scrub at one point in time. Whether it was because of a predator or infection, we don't know for sure, but the shell was completely intact pretty much when she found it, so we aren't sure exactly what caused that death. It could have been infection, but there's still a lot more to learn about the potential infection and pathogen pathways between these turtles, as well as between them and gopher tortoises, given how much of the habitat they occupy together. Now, this is kind of the cool stuff. So I'm gonna tell you some groundbreaking news that we found in this section here, which is really kind of cool. We have observed breeding of these turtles, and basically every month we've been out in the preserve. The only exception is we didn't see any breeding in January, but we know there's got to be breeding in January. But we basically observed copulation, or at least attempts at copulation, by male turtles of female turtles in every month of the year. That's a big thing, number one, because in other parts of the range, there's winter. They don't have the opportunity to breed throughout these times because they're inactive and burrowed underneath the ground. In fact, they breed so readily that these are actually 2 that we were in the middle of trying to apply radio tags to, and they just couldn't keep off each other and started breeding in the box we were holding them in. They just couldn't leave each other alone. So they are all about breeding down here, which is pretty interesting. So from August 2019 to 2020, we documented eggs in May, July, and August, kind of the typical spring summer period that you'd expect box turtles to nest in. But this is where the big news comes in. So even after that first year of radio tracking that we documented, we applied 6 more transmitters to 6 of the female turtles in the preserve specifically, so we could catch them once a month, take them to a local veterinary clinic, the Naples Humane Society, and get them radiographed to check for eggs. And one of the coolest new things we've found is the earliest ever documentation of gravidity, which means to have eggs, in box turtles of any species anywhere. We documented Priscilla, our girl, as having eggs in January. That's big because no other box turtle populations could be documented as having eggs in January because they're all underground hibernating / brumating at this point in time in their life. They're not going to have eggs if they're under, a foot of snow in some places in January, right? So discovering this is huge, and this is going to be a really great paper that we're going to write up on this early reproductive effort in box turtles. Then we found eggs in February as well as eggs in March, and we're going to take them back in for radiographs in April. I'm sure we're going to find more eggs in April. I'm sure we're going to find eggs probably in almost every month of the year, excluding kind of a slow period around September through December. There's about 4 months they seem to kind of take off in terms of reproducing eggs, just probably because they're so well spent on their energy in terms of producing these clutches. Their clutch sizes range from anywhere from 1 to 5 eggs, and we're going to get some really good data to look at, see if that clutch size kind of decreases as they produce more clutches. This is what those radiographs actually look like. If you look closely, you can kind of see the outlines of the eggs. And in some of them, it's like, man, how are there so many eggs in that tiny little turtle? And how do they pass all of those eggs? But the good thing about this is their eggs are very soft, very malleable, so they're very easy for them to pass. So they can pass even some of these largest eggs right out through their cloaca. And some of them have four eggs here. 3 eggs kind of seems to be about the median or the mean of clutch sizes, but they can have anywhere from 1 to 5 eggs. The average egg size is about 34 millimeters. So if you put that into inches, that's a little bit about 1.5 inches approximately in terms of how long they are. So they're pretty long eggs. And so far, we're seeing a nesting interval, at least starting in January, of 1 to 2 months. Priscilla, for instance, nested or had eggs in January, didn't have eggs in February, but now has another clutch of eggs in March. So she had eggs in January, laid those eggs, then started working on another clutch in February. We didn't see them yet, and now has them again in March. And we know for a fact that in past months, Priscilla has nested in May, and she's nested in July. So we're likely going to see at least 4 or 5 clutches from these animals. And if we see a clutch size of 5, that's going to be another record for the most number of clutches ever documented in the species, which would be really cool. And then one really cool thing is we have one individual, Missy. She had eggs in February. She had a clutch of 3, then had a clutch of 4 in March. So she's clutching within a month of herself. She has enough energy to lay a clutch, pop out another clutch in between those months, which is just outstanding and amazing. And it's going to yield some really good data and probably a paper that's going to have a lot of citations in its time. And we're noticing at least 2 per season, but I can almost guarantee it's going to be closer to 3 to 5 in terms of number of clutches, which is, again, just ridiculous reproductive activity. They've documented them laying up to 4 clutches in Everglades National Park. But the earliest they ever documented them nesting in Everglades National Park was February. So we still beat them by an entire month with our January samples. So we got the record, which is really cool. But with that kind of gives the general take home of the data that I have for you. I'm happy to take any questions, but I always like to point out this is not a solo project. It may have been kind of my brainchild, but without all of my students here, and these are students that are currently in the class or students that have been here previously. Adrian Rodriguez, who's here, give Adrian a shout out and a round of applause. We've got Julie Rodriguez, Julie Laredo, that is here as well. Give Julie a shout out. We've got Cody Weber. We've got, I think Christina was here, or at least she was here, and I saw Crystal here at some point. They were all here in the class, and they do so much good work and continue to do great work for our research project and for the turtles. So wouldn't be able to do the project without them. I also like to thank my lovely wife. She is also in the chat right now. She's been my field buddy a couple of times, especially last year when everything got locked down. I wasn't able to bring students out. She graciously came out to the field, helped me track, helped me measure and monitor turtles. So wouldn't be able to do it without her help, and also her patience of allowing me to bring turtles home to take measurements and to take data from them and keep them in our spare bathroom where they graciously cover it with poop while they're being held for tagging purposes. So thanks again, Babe. I appreciate that. But we also have a lot of connections with, again, a lot of other conservation organizations. We're working directly with the Sanibel Captiva Conservation Foundation, Chris Lekowitz and Mike Mills up there. They helped us with our initial tagging efforts of these animals. They also are going to help us with some publications. We're going to compare data between our populations. Brooke Talley and Brad O'Hanlon at FWC, they took particular interest in our project and have kind of folded us into the overall monitoring programs of turtles for the state of Florida, which is really cool news. They help us both financially as well as scientifically, which is great. So we're very, very happy to have them. And we wouldn't know all this really cool egg data stuff without Dr. John Morton and all of his awesome staff at the Naples Humane Society, who cut out portions of their day to radiograph sometimes between 6 to 10 turtles for us. Even though they have a bunch of dogs and cats that need treatment, they take the time to radiograph our animals, and they don't charge us a dime, which is absolutely amazing. It's really, really awesome of them to do that. So we are very thankful for them. And if you have dogs and cats and you want to see a good vet, I'll advertise for them. They're the best. They even treat some of my own pet turtles, so he's good with exotics too, which is great. But if you have any questions, now's the time to ask them. I always put up here, if you have an Instagram page, we are the Southwest Florida Turtle Project on Instagram. Feel free if you want to see lots of cute turtle pictures. And I always put this up there. If you have a cell phone, you can take a picture of this barcode, and it takes you to a GoFundMe page. If you feel the urge to donate and help us support this project directly, you're welcome to do that, and we'd appreciate it. But at this point in time, I will take any questions you may have. Any questions, or was I that thorough that I just a masterful teacher?
Thanks for the shout out, Babe.
No problem, honey.
I'm very proud of what you and your students achieve and your dedication that you all have to go out in the fields and get messy all the time. Do you think the preserve is, I guess, grossly over-inhabited with box turtles at this point, or do you think it's a healthy mix?
So the population size is actually quite small there. We've only tagged about 20 unique individuals. We know they're laying lots of eggs, but we just don't have any real encounters juveniles. I think we found like 2 or 3 actual juveniles. I don't think there's a risk of like population overabundance there, because we think the little ones, when they hatch, they can actually probably fit through the fencing and probably disperse to other places. I mean, there's probably a few that are going to build up home ranges inside the preserve, but I don't think there's a gross over inhabitants. We did find them interacting together, but compared to some of the other sites that we go to, the interactions were basically exclusive to mating. Whereas one of the other sites that we see, we find like males all huddled up together all the time. We found males fighting with each other and dominating each other at other sites. We haven't seen any of that at the preserve. We've just basically seen them together during mating time. A good question.
Hey there. On the topic of
you don't get to ask questions, Adrian. You have a paper to write.
Talking about juveniles and females nesting and everything. Are we in like pretty good suspicion that there's a lot of predation going on at the preserve?
So you would think, especially with someone like Becky, who's out there all the time, that there would be more evidence of it. I know she has found some box turtle nests predated. She's actually found some successfully hatched nests as well. But you would think there would be some more evidence, at least seeing the eggshells and things like that, that there would be predation. But we know there's a lot of raccoons, foxes, and coyotes out there, so there's no shortage of predation pressure. So that's a possibility as well. But if these things are laying 4 or 5 clutches a year, if they were getting predated, I think there would be more evidence of us seeing the predated nest than maybe we are. But of course, we don't know exactly where they like to nest either. So maybe they're nesting in the thick middle of a bunch of dense scrub and smilax that we can't get to and don't really want to get to. And maybe we're not seeing the evidence of those predated nests anyway.
Yeah.
Any other questions? Awesome. Great. Everybody have a great day. Thanks for coming.